Okay, let's start off the today's class. If you can hear me, I ask you raise your hands. Then I can see my voice is working. Okay, so it's good. Okay, so let's resume the topics from data modality, the part 2. Okay, so let's continue to talk from the VCF format. So we discussed about the how we call the variant from the sequencing data set, and we get the sequence raised from the machine, then map to the alignment and put it into the SEM or BEM format. Then out of these files, we call the variants and we identify whether the samples has the mutation or not, right? So in this process, we also use the very specific format called VCF. So VCF is the it's very particular format for the variant calling. Sorry, one second, please. Sorry, guys. Sometimes the calls from the university they accidentally came in my office, so sorry for that. So we use very specific format called the variant, the VCF format, and this is the shortly the representing the variant code format. So it's just shortly VCF. The motivation for using this VCF file is the same as we developed the SEM format. So people want to develop certain format to represent the variant or the genotypes we get from the samples, right? So people has to come up with the certain standard to look up the variant from the large amount of data sets. So VCF is the one they come up as a solution at the first time. And again, the 1000 Genome Project, the working group, they designed this format to store large number of genotypes they get from the 1000 Genome Project and distribute to the other researchers. Then people can access their data set, and also people all working in the same condition because we basically use the standard, right? So we say from 2011, people conventionally use this format, and nowadays I mean without VCF we cannot do any, types of the genetic analysis, because this is the basic things for any types of the genomic data sets. So let's look at what VCF contains in their format. So VCF is basically the file format based on the matrix type. So it's basically containing the list of variants we got from the samples, and then the columns representing the information about the variants and samples, right? But if you look at the VCF, your first glimpse on these files is actually the headers, right? So if you open up the VCF file, it has the long lines of headers in the file, and the headers is like, the lines start with the char string, like a hashtag, right? So the first line is the file format equal VCF version 4.0. And the second line is the file date, right? So 2010 and 77, and the source and reference and all of that information. So what VCF header contains is, this is the sort of the commentary of how they made VCF in their analysis, and which, the version of the reference genome they used in the data set, and what kinds of information they store, and we can find from the VCF file. So these comment style lines is one of the conventions people used in the computational science. So if you store a certain data set, people always put their notes or their tags in their file. So not just the VCF file. If you work in a computational science, if you work in a certain, the files from other purpose, they also can leave their comments in their file, and VCF follow basically that notion. So if you think about like an Instagram, you put your photo onto the Instagram, then underneath that part you put the description, and underneath that description you put the hashtag, right? So hashtag is basically like the miscellaneous information that can you want to add more stories or more information onto your post, right? So this is the same as we can find in a computational science or genomics, but we using the first few lines of comments instead of the tail points, right? So you can also leave the tail points at the end of your file, but normally conventionally people put the comment lines on very first line of your files. And VCF2, and what these comments do, readers can use, or users can access that information, but also computer look up this information and check whether the useful information the computer used in their analysis, and VCF headers storing sort of this information, and we can call it sort of the meta information for the VCF, right? And this is the header part. And after the header part, the end of the header part, it has the column names, right? So these column names start with only one char string, right? So only one the chart tag. And it starts with the first column, that's a chromosome, and the second column is the position, and the third column is ID, right? And the reference, alternative, and the quality, and the filter tag, and information column, and format column. So how many columns? 1, 2, 3, 4, 5, 6, 7, 8, 9. These 9 columns is the mandatory things we store in a VCF file, right? And after this 9th column, then we put the columns for the samples, right? So if you do only 2 samples in your analysis, then you add 2 columns in here. But if you have like 1,000 samples, then you have 1,000 columns after that, right? So what this 9th column do is they store the information describing the variant, also describe the genotypes from the samples, right? So we are following that information from the next slide. So this is one example. So you find the mutation on a chromosome 7, right? And then this position, and this is about like a 98 base pair window, right? And one base pair, you can look up the mutation, and on this locus, you have the mutation, which is the A changed to Z, right? So that person has the A to G mutation in this locus, and this person's mutation has a heterozygous genotype, right? So you can read that some of the reads are A in their read, right? And the rest half of the reads, they have the G alleles in their alleles, right? So you can see that the 50% of the total reads are A, and other 50 is the G. So we can say this is the heterozygous genotype situation, right? Okay, so how we can store that information into the VCF file, right? So if you look at the VCF file, we first put the chromosome position, right? So this is the chromosome 7. You can call it chromosome, or you can also call it contig, right? So why we have two different notation? Because the contig is more mundane, so it's more old terms than chromosomes, right? So if you think about the days of the Human Genome Project, people building the reference genome using the concept of contigs, right? So from the time, people normally call the chromosomes contigs, right? So contig 1, contig 2, but more modern context, we call it chromosome 1, chromosome 2, right? So sometimes you might find confusing because some situation people call it chromosome 7, some people called it's a contig and it's a chromosome 7, right? So but anyway, the first column is chromosome 7, which you can find in this screenshot, and the second column is the position. So position is the 117175373, so this is the position, and the third column is the ID. So you can put the identifier from certain database. So if you think about the database storing the SNP information, we use the DB SNP, right? So you learned from the session 3. So that case, you can put the ID from the DB SNP database, right? But that case, they don't have any identifier in this position, so we can omit that. And the fourth column is the reference allele. So reference allele is the A, right? So this is the same A on here. And the alternative allele we store in the fifth column, and that is the G, right? So we can clearly see that the position on this person has A to G mutation, right? And how good this variant code is, then we can store the quality information, which is the 90 here. And the next column is the filter. So whether that is the past the VQSR tag or not, right? So sort of that information we store in the filter column. And info column is the description of the quality related to this variant. So you can put any kinds of the quality matrix, quality information into this info column, right? And the format column is describing the genotypes that this sample has, certain sample has in their VCF, right? So format has the only representing the notation on here. So instead of including the actual information, it actually including the notation. So how they gonna represent the genotype in a sample column, right? So format column has the GT here. So GT means the genotype. So that is the equivalent to the information on here, right? So this is the sample ID. So first sample LF1396, and this sample has the GT information, which is the 0/1. So that means that heterozygous genotype. So you can think about like this is the diploid situation. So the first allele and the second allele, right? So the first allele is the 0, right? So 0 is equal to reference allele, so on here. And the 1 is the alternative allele, right? So this is the 1 on here. So why this is not 1/2? This is basically based on the computational notation. So computers start counting the numbers not from 1, it starts from the 0, right? Most of the computational language actually start from 0, right? So that case, they follow the computational notation, and that in the sense, we can say the 0 is equal to reference, and 1 is the first allele in an alternative column, right? So you can put it in a different context. So let's say this person has the 0/0, then we can say this person has a reference homozygous genotype, right? But if that person has the 1/1, then this person has the alternative homozygous genotype, right? So this is the basic ballpark. Okay, so if you look at here, then this is again the same information. So 0 and 0 home ref, 0, 1 head, and 1/1 is the home alt, right? But some of the genotype column, it can have the dot/dot. That means the unknown, right? So then the question is why we don't have the genotype information in this person. The reason is that this position and these samples doesn't have enough data. So data here is the sequence read. So we cannot read any genotype information from this locus, right? So if you look at certain position, like in here, this is just one example. So if you read this position, then this is the two individual, right? So the first individual is mom and the second individual is the kids, right? And you can clearly see that these two positions, you can read the heterozygous from each person, right? But if you read that position on here, then you can read the genotype information from kid, but you cannot read the information from mom, right? So that case, you can say, if you read this position, then you can say kids probably have 0/0 because there is no mutation in here. But if you look at mom, then you cannot judge that, this locus mom has the heterozygous or homozygous genotype, right? So you just put the dot/dot because you are unknown on this position, right? Okay, so and this is the meaning of the unknown genotype, right? So back to the this screenshot. So in the VCF file, you can represent the genotype from single sample, but also you can put multiple samples, right? So here you put the two samples, right? So your first sample has the mom and the second sample is the kid, and both samples has the heterozygous genotype. So you put the 0/1, 0/1, right? And if you have that sample, then you put the that column and you put the genotypes on here. And if you have the 100 samples and you put 100 column, then you put the genotypes, right? So in the multi-sample VCF, what you have to understand is the differences between the variant and genotype. This is the key things you have to understand in the VCF file. So the information about the variant and genotype, I mean, it sounds quite similar to each other, right? Because the variance is also changes of the nucleotides in a DNA, and variance is also the changes in a DNA nucleotides, right? But variant and genotype is quite, different meanings in the context of the VCF, right? So the variant is the information describing in a locus, right? So if you look at here, the variant is the chromosome 7 and this position, so this locus, and the variant is the C to T, right? But then genotype is the belong to each individual, right? So in this examples, mom and kids, we have the two samples, and these two samples has the same genotype, right? But mom could have 0/1 heterozygous, and kids could have 0/0 reference homozygous, right? So that case, you can say that the mom's genotype and kids' genotype can be different, right? So these examples, the genotype between the mom and kids are same, but the certain variant position, certain locus, they have different genotype across samples, right? So you have to understand the information variant and genotypes can be different, right? So that is the one thing. And another information is we can calculate the allele frequency, right? So this info column contains the allele information, and this AF is the shortly called allele frequency. And this is the frequency of variant allele in this position, right? So allele frequency is 0.5. That means that we have 2 samples, and out of these 2 samples, we have 1 variant allele in a mom and 1 variant allele in the kid, right? So out of total 4 alleles, because we have the diploid and diploid, so we have the total 4 alleles, and we have the 1 allele for the variant in each person. So we sum up this 1 and 1, so 2 divided by 4. So we can say that this is a 0.5, right? And if you have, mom has the heterozygous genotype 0/1, and if the kids has the reference homozygous genotype, then that is the 0/0, right? So that case, the allele frequency is going to be 0.25, right? Because it's 1 out of 4, right? So if you have the 1,000 samples, and if you have only 1 person has 1 variant allele out of 1,000 people, then you can calculate 1 divided by 2,000, and you can calculate the allele frequency. Okay, so, and this is the one, the global screenshot of the VCF format. And as you can see here, so this is the header here, and this is the, each lines represent the individual locus, right? And individual locus has the variant information. So what is the reference allele and what is the alternative allele, right? And as you can see here, you can store the SNP information, SNV information in here, but you can also store the indel information in here, right? And also you can store the biallelic situation, which is the, in a one single locus, you have only 2 alleles, right? So in this locus, you only have the 2 types of alleles, G or A, right? But some of the locus could have more than 2 alleles, right? So it could have the A allele, G allele, and T alleles, right? So in that case, you can say that this is the multi-allelic situation. Okay, and also you have the index here, right? So this is again multi-allelic situation. Then this also has the deletion on here. So one variant could be deletion, right? GTC changed to G, right? But the other person could have the insertion. So GTC, but it changed to GTCT, right? So you add a T because there is an insertion happening, right? So multi-allelic has following the computational notation as well. So if you look at mom's genotype, it has the 1 and pipe and 2. So I'll tell you the differences between the pipe and slash, but back to later. And if you look at the 1 and 2, so one allele has the 1 and the second allele has the 2, right? So that means that 1 is equal to G, and the second on here 2 represent the T, right? So mom's genotype should be G to T, right? And that genotype has the T to G, and kid's genotype has the TT, right? And this line, the insertion and deletion line, the mom has the deletion because GTC and G, right? So we could say this is the heterozygous deletion, right? And that has the heterozygous insertion, right? So 0 is the GTC and 2 is the GTCT, right? So that has the, one strand has the insertion. And kids has the GG, so kids doesn't have any mutation. So kids has the, kids has the mutation, kids has the alternative homozygous SNP, right? So very complicated, right? But, this is the sort of the real world situation, how, individual has their mutation in the genome, right? Okay, so, and the next point is what is the differences between the slash and pipe? So pipe represent which strand this mutation located, right? So we have the 2 strands in our genome because we are diploid, right? So if you use the specific algorithm to track down where that mutation from which strand, right? So that case, you are using the algorithm called phasing, right? Phasing is the algorithm to detect where the mutation located in a 2 out of the 2 chromosomes, right? So why phasing is important? If you look at the kid's genotype, then you can track down where these alleles coming from, where it is from mom or dad, right? So if you look at here, then you can see that this is not, yeah, this is true. So if you look at 1 and 1, then you can easily track down this is from the dad, right? And this is a 0, 0, and that actually from mom and dad. And this is from, again, mom and dad as well. And this is from 1, 1, and that's probably the wrong genotype, right? So that case, we are using the phasing, and we can check how this variant transmit from parents to the kids. So that case, you are using the phasing information. Okay, so this is just very brief examples of the VCF. It's like a tip of iceberg. So if you look at the VCF file from the patient, the real data set, you have like 10 millions of the lines from the 10 samples or 1,000 samples or the 10,000 samples. So you have to deal with that large amount of information, with certain computational framework, not by your hand, not by your Excel, MS Excels, or not by your, the manual lookup. So, we have to come up with certain, solution to look at all of this large number of the genotypes and put it into your analysis. We are continue to discuss the issue in the later section. Okay, so there are some key terms in VCF. So headers contains the meta information for variant calling process, and there are run command for bioinformatics tools for mapping and variant calling. And one of the program, the famous one, is the GATK I showed briefly on previous slides. And also header contains the version of the reference genome and also run data as well. And if you look at the chromosome, we sometimes call it contig. And also the second column is also the very important information. And we put the positions of the variant, right? And this position is based on the one base coordinate, right? And why I explain this, because VCF format has the one base coordinate, not the 0 base coordinate. This is very very funny situation. I mean, I laugh when I feel tragedy. So we have the devil coordinate system in genomics. So you have to embrace this sad situation. So we have the two different coordinate systems. So when you represent the position or location of your mutation or gene or transcript, you can represent the 0 based or 1 based. So 0 based is we call the half open. So we have the mutation happening here. Then you say this is the 8 to 9, right? So this is somehow between here, right? So when you represent it like an interval, so like a range, then you normally use the 0 based position. But mutation is different, right? So mutation, we represent, if the mutation happened in here, we describe this as 9 to 9, right? So this is just a single point. So on here, right? So the reason is that when you think about the position, so if you have the sequence like a TACG TCA, So you can say this is the 1, 2, 3, 4, 5, 6, 7, but also you can say 0, 1, 2, 3, 4, 5, 6, 7. So if you're based on the zero-based position, then you have to put this T position has to be on the 0, right? But if you're following the one-based coordinate, then you say 1, right? So this is quite, a nasty way to represent that information, but all of the genotype, the genomic information, not just by the mutation. So it can be represented by the intervals, represented by the certain area. So people use both systems, so zero-based and one-based. So VCF used the one-based system. Okay, so in here. So this is the critical information for your learning. So VCF used the one-based system, right? So full closed the coordinate, but like certain position like a BED the format or BEN file, right? Or BCF format, they use the zero-based position, right? So because they are generally describing the range or intervals, right? So this is the differences between the BED between the zero-based and one-based, and each file format has their own preference for the coordinate system. So you have to understand, the lots of genomic files, they have different coordinate system, right? And this is always confusing, so you have to look up, but some point you will be quite familiar with that system. Okay, so position is one-based coordinate, and the VCF has two types. So one is the individual sample VCF, that is only you do the genotyping for only one sample. And another format is the joint VCF, or we call it multi-sample VCF. So literally in a VCF file, there are large number of samples in the VCF, right? So VCF, multi-sample VCF, we do the genotyping for multi-samples, right? So why we have to do the multi-sample or joint VCF? The reason is that if you include more and more samples, then you have better quality for variant calling, because it creating the likelihood model for quality distribution. So if you want to do the genotyping for only one sample, you cannot compare the quality of the variant, your samples to something, right? Because only one sample, right? But if you have multiple samples in your VCF, then you can basically compare the quality of the variant between the samples, and you're creating the backgrounds of this quality distribution. Then you can judge that sample's variance is very poor quality. So we can judge that is the probably non-pass call in the VCF. So this variant information, the multi-sample VCF has the strong advantage for the quality-wise usage, and so lots of people wants to use the joint VCF for their analysis, but also in a clinical situation as well. And also, more important thing is we want to find the reference genome in all samples, right? So why do we care about the reference genotype? So back to the trio situation, this is the one example. So if you want to find the de novo mutation from the trio, so let's say you're a doctor working in a pediatric, and you see one patient, and that patient looks like developmental disorders, right? And then you want to judge that, you want to evaluate which mutation happening, which gene in that patient, right? And you want to find out de novo mutation in this patient, right? And you have the two options. So you can just the genotyping that patient, or you can do the genotyping the kids plus his or her parents, right? So if you do the single genotyping, the single VCF, then you can find the heterozygous genotype or reference genotype or homozygous genotype from the kids, right? So kids who are, suspect to have the developmental disorder, right? And if you think about the de novo mutation is the heterozygous genotype, right? Because the de novo mutation newly arising in one of the parents' germline cells, and that is the transmit as a heterozygous genotype, right? So you can find lots of heterozygous genotype, but you cannot judge that which heterozygous genotype is the de novo mutation, because we have billions of the heterozygous genotype in a person, but de novo mutation only occurs in a 70 events in a single person, right? So you have to look up the parent genotype, and parents should have the reference genotype in their genome, right? So that case, you can confirm that that locus doesn't have the mutation exist in a parent, right? So there is no mutation, and the kids has the heterozygous genotype in the locus, then you can judge that, okay, so this mutation probably de novo mutation, right? So in that case, we are taking advantage of having reference genotype in a multi-sample VCF, right? So if you look at only single sample VCF, there's no way we can judge that whether other person has the reference genotype or not, right? So you have to come up with the multi-VCF genotyping, then look up all people's genotype in your analysis, then judge the variance scores of your interest, right? And this is one example for the de novo mutation. But if you want to compare, like, if you want to do analysis of, taking medicine by genotype, that case you want to compare the reference genotype, heterozygous genotype, and homozygous genotype, then you want to compare the additive situation, because additive working as a, you add a one allele to each person, then you will see increase of medication or decrease of medication, right? So, think about the additive situation. So you add up the one allele or two alleles, right? So reference genotype doesn't have any alleles, variant alleles, so it's a 0, right? But if you have the heterozygous genotype, you have the one allele, right? Homozygous genotype, you have the two alleles, right? So you can say 0, 1, and 2. So you will see linear relationship of the genotype and you're taking medication. So either it can be increased or decreased, right? So that's that situation. We're also considering the reference genotype in your analysis, right? So reference is critical. Then what is the disadvantage of using joint VCF? If you want to do joint VCF, it's computationally very slow and also intensive, right? So if you do single sample, then you can only look at one samples at a time and evaluate the variant codes. But joint VCF, you take up multiple samples, and also you have to look up each individual's reference genotype in each time. So if you think about, the session 3, if you probably remember the increase the number of the rare variants in 1,000 genome project. The reason is that rare variants is very unique in each individual. So if you're including all the rare variants in a samples, then you are exponentially increase the number of loci in your analysis, and that is the happening in joint VCF as well. So if you do the joint VCF work in your analysis, it's computationally very slow and very intensive, right? So that's why we have to use lots of CPUs and memory for your analysis. And also another practical issue and very famous issue in our field is N+1 issue. So let's imagine the situation, you analyze the 2,000 the patients from your clinic, right? And then after a month, you have one more patient, right? And if you want to add up this one patient to these 2,000 patients, then there is something, right? Because you already create 2,000 patient VCF with the long hours, lots of computational effort. But at the end of your analysis, one more patient add on, then you have to start from the first, right? So that's why people are quite, wants to develop sort of the smart way to including these extra samples in a joint VCF situation. But I mean, this is one of the well-known, the problems. People still want to improve the joint VCF work in a genomics field, right? Okay, so and let's look up more details of the columns. So we have the two columns describing the variant and genotype quality. So first column is the info, which is the variant level information. So in the case of joint genotyping, and it will summarize the variant level information, and it will summarize the information from multiple samples. And it's describing the characteristic of variants codes you find in a calling process, right? So it includes like quality matrix like a depth or mapping quality, allele numbers or allele counts. So let's look at the depth. Depth is literally how many times this locus covered by sequence read. So 10 reads or 100 reads or 1,000 reads. So that's stored in a depth. So if you see higher depth in your VCF, in your variant, then that represent the generally high quality variants in your analysis. And mapping quality as well. So higher mapping quality, that literally high quality of your mapping happening in an alignment situation. And allele numbers is the total number of alleles you identify from your multi-sample analysis. So if you analyze 10 samples, that will have 20 allele numbers because we are diploid situation. And allele count is the number of variant alleles from the from each genotype. So as we see in these examples, we can add up the variant allele numbers from the VCF and store in an AC column. And we also have other quality information, which is the like a strand-odd ratio, which shortly called S4-4. And this is the representing the situation of how the sequence read are biased by the PCR implication or not, right? So either reference allele or alternative allele is the bias toward the PCR amplification. So that case, if there is no bias in the sequencing, then the reference and alternative allele should have the equal numbers in their sequencing for the heterozygous genotype. But if there's any bias towards alternative or reference allele, then in that case we can calculate by the statistical method like a Fisher test or chi-square statistics, right? So we are using the same the 2x2 tables, which is the reference or alternative alleles, and either it's be the fourth strand or the reverse strand, and that is the whether they had large, the fraction of the GC counts or lower fraction of the GC counts, then that will whether they will affect on the quality situation. Then in that case we can use this matrix to represent whether this variant is severely affected by the PCR amplification or not, right? So that case, people use this matrix. So other than these 5 matrix, we have more quality matrix, but I only put the very famous one in here. And in this info column, other than quality matrix, we also have the annotation information. So annotation is the description of the variant consequence. So either it's be how many times occur in your samples or in a population, and which gene this variant locate, or which transcript this variant locate, or what kinds of the variant consequences belong to this variant, right? So if you only have the variant information like in here, then you only have the genotype, you only have the allele information, right? So, and also the locus, right? So where these variants happen, right? Like a chromosome position and the allele changes. We don't have any information about which genes or which transcript or which variant consequences happen by this mutation, right? So you have to calculate that information from certain program, right? So we're coming at the back. And another column is the column called format, and this is the genotype level information, and this is the information at the individual level, and this is the number of columns are the same as the number of samples in the analysis, right? As we see in these examples, right? And this is the describing the characteristic of the genotypes you found in a variant column, right? And it has the information about GT genotype, and it has the information about allele depth, right? And it has the information about depth, and it has the information about the genotype quality and genotype likelihood, right? So the differences between the allele depth and depth. Allele depth is the depth of one allele, right? So if you have the two alleles, you will have the two information for the both alleles. And just depth is the total number of these AD information. And the format color indicate the list of items. So format columns doesn't have the consistent constitution across the variants, right? So each variant has their own format information, like like in here. So the first line could have the GT, ADDP, GQPL, and the second line, which is the next variant, could have the only 4 items, right? So if you look at here, this is the same. So the first variant has the 4 items in a format column, but the last one has only 3, right? So this 4 is constituted in here, and these 3 constitute in here. So this variant, this line doesn't have the HQ in their format, so this is not existing in the samples information. So format basically use this key information linking to each individual's genotype characteristics, right? So this is the differences between the variant, the info column and format column. So info column is the representing entire samples information across here, but format is only indicating each of them, right? So this is the different situation. Okay, so let's look at how we do the variant annotation. So variant annotation is the process to assess the consequence of the variant from your variant calling. So why we need this? Because variant calling only does genotyping, and it does not tell you what the variant would be, right? So where the variant located in a coding region, or whether variant alter protein coding sequence or not, or which gene variant located in, right? So variant annotation uses the reference data set for the transcript, because mutation can be evaluated by each transcript, right? And also transcript sequences. And if you want to do variant annotation, you can use, there are many options to do this analysis. So you can, one of the famous few things like an ensemble VP or ANOVA or SNPIF. So if you go to here, or if you go to here, then you will see how these tools are actually working, right? So variant effect predictor. So VP is shortly the variant effect predictor. So you can put your variant information here, like you can put like this, and you can see how the mutation is changes in a reference data set, or you can use the other tools like an ANOVA, right? So I want to do with you some exercise about how we actually do the annotation and how you look in the analysis. So we can try VP. So I pick up one variant, which is the chromosome 3 and this position. So we can basically copy, drag, and this, and put it on here. Okay, so I don't have to copy everything. I copy. So chromosome 3 and the position in here. Then C to A. So that will be see. So this is the VCF format. So chromosome position, ID, we don't have ID. So, and the reference allele C, and what is the A allele? This is the variant allele, so A here. And we don't need this because this is the variant level information. Then we can click run, but also we can click just the instant run, right? Then we'll see what this variant is, right? So this variant is in the PPAR gamma, so PPAR gene, and this is the mutation called missense variant, right? So this is changing the protein sequence, right? And few of the predictions, like for the whether this mutation is very bad for your health or not, then this algorithm show you, okay, this mutation looks deleterious, so something bad, or probably damaging, right? Okay, so this is very, brief run for the mutation. If you run it by yourself, then you can click this run, then you will see entire information about this mutation. And one spoiler, this mutation is actually the mutation found in a type 2 diabetes patient. Then you can, this is one of the famous mutation in the type 2 diabetes. So you can actually see how they will represent in an annotation situation. Okay, so there are some resources for the variant annotation. So you can use any types of data set that represent the characteristics of the variants. So it can be protein sequence, or it can be gene transcript annotation, microRNA binding sites, because sometimes it matter in a biology, and transcription factor binding site, because it's critical in your development, and active transcription regions. So if you want to look at patient's genome with the Alzheimer, then you want to see the genes are actively transcribed in a adult brain, amygdalas or hippocampus, then you can see those regions and find the mutation related to that gene. So any types of the resources, then you can describe the variant, then all of that information can be stored in the VCF file. And each data file has different formats. So as I show you briefly in the previous slides, then in a genomics field, we have all of the different formats to represent the genetic information. So one famous file format is the gene annotation. So in our genome, we have about 20,000 genes for the protein coding. We have about 40,000 genes for the non-coding genome, as we learned in the previous section. Then those information has to be stored in a certain format. In that case, we use the GFF3 or GTF format, and that is look like this. So it's a long one here, GFF. Oops. Okay, to GTF. Oops. GFF3, where are you? Not on here. That case, you can come to my space. Let's share my computer. Oh, where am I? Here. Oops. I'm already on here. Oops. Oops. Okay, so let's look at the GTF file. Oh, sorry. Sometimes my computer is not working as expected. yeah, it takes time for the Dropbox syncing. Dropbox used to be good, still better than Google Drive, but it introduced very brainy system, smart things, so they don't work properly. Please bear with me. It will be quick. Okay, we can go different space. Go away. Okay, so let's look at the GTF file. It actually looked like this. This is the short one. Okay, so this is the GTF file. So this is the file from the GenCode database we see previously. And GTF file also has the header because this is the commentary and this is the basic information about the GTF constitution. And then you have the each gene's information on each line, right? So gene has the gene ID here and gene type and gene names as well, right? And I mean, this gene called DDX11L1 located on chromosome 1 and the position, right? So using this information, then you can evaluate where your mutation happened in this gene or other gene, right? So you have the position information, then you can easily track down where the mutation happened in a certain position, right? So this is the one way we can, using the gene annotation for the variant annotation step. This is the GTF format. And also we have the region annotation information. So if you think about the chromosomes, so not all the chromosomes are open in a certain cell or certain activities, right? So if you develop in a your kidney in a maybe 2 years old, then that time the cells and genes related to the kidney development has to be expressed, right? So that chromosome area has to be open in the time, right? So if you measure that area in a 2 years old kidney samples, then you can evaluate your mutation is open in the position or not, right? So that is the linking information between your mutation to the certain biology, right? So one example here is you can find from the genome browser. So if you go to genome browser, so we have the region of SN2A, right? And you will see transcript information in here and another transcript information in here. And you have the information called encode CCRE. So encode CCRE is the cis regulatory element. So cis regulatory element is equal to transcription factors. So you can simply think that this is the transcription factors, what you learned in molecular biology. So what you want to know is where these cis regulatory element or transcription factors are open up in a SN2A region. So this transcription factors binding sites is not, happening all of the, the nucleotides across the SN2A region. It actually happening in a certain position, right? Like in here, like in here, like in here. Each one should have the different information, right? So transcription factors called E0 E to 0 blah blah blah, and that is the happening on here, and this regulatory element happening here, and this element happening here. So if you measure the information from the previous experiment, and then you can link that experiment information to your mutation profile, right? So if this region is critical to brain development and the mutation happening here, then you can easily think that that mutation is probably bad for the brain development, right? So this is the one way we can evaluate the variance. So that's why we are doing the variant annotation. Then we can systemically evaluate how this mutation will affect on your biology, right? So this is the one way we can use that information for the regional annotation. Okay, so last step of the VCF information is the very specific program called HAIL, right? So HAIL is the latest genomic analysis framework. So if you want to analyze the large amounts of genomic data set from the patient, then you have to use this program, right? So, if you think about the VCF file, and if you want to analyze the 2,000 samples from the whole genome sequencing, and the output file is about 2 terabytes in a compression. So if you decompress the file, then it will be like 10 or 20 terabytes. And there is no way you can, upload these 2 terabytes, information to your memory, right? So, if you think about the Excel situation, if you want to open up Excel file like 2 megabytes file, then you can easily open up in your laptop, right? But if you open up the 200 megabytes Excel file, your computer, basically sick, right? I mean, computers say, no, that's not going to happen, right? So if you want to analyze 2 terabytes compressed file in your computer, then memory can handle that, right? So some smart people in the old days, like around 2010, 2012, that they was thinking about how we dealing with that situation and how we prepare future genomic analysis. So these 2 brilliant people from MIT and Apache Spark, one of the big company in the data science, and they are coming with the solution called the HAIL, which is based on the Apache Spark Hadoop system, which is the developed for the large scale database. So if you think about like big data or whatever the metadata in these days, people use the Hadoop or a Spark system. And the geneticist in an MIT, Benjamin Neal, he collaborate with the Spark team, and they develop very specific program only for the genomics, and that is the called HAIL, right? And that is also implemented in a Google Cloud or Amazon web service, so people can use in a cloud system. So in a not near future, in present days, and if you work in a genomics field, if you work in a biological data science, people can easily access to the cloud computing and the analysis, the large amount of data set, and it will change your lots of the analytic frameworks, and all of these things can happen by this brilliant advancement. So how HAIL is working? So if you do the variant calling process from your sequencing, then you will have the VCF file, right? So using this VCF file, you kind of handle in a Spark program. So you have to load that VCF and transform the VCF into the certain format, then the HAIL can read it. This is called matrix table, right? And HAIL convert the VCF file into the matrix table. And this matrix table, we called it genomic data frame class, right? Why we called it very specific format? The reason is that if you look at the VCF file has very multidimensional, the information is stored in a single file, right? So if you look at the columns, so columns on here, this is the sample information. So sample information has to be linked to their phenotype, their age, their sex. So all of that information you have to retrieve to the VCF situation. So one matrix stored in a column, and the row table is the variant, right? So the variant is the each lines, and is the store as a row table. So that is the second, the dimension you can identify in a VCF. And the third file is the entry table, which is the genotype information, right? So you can summarize the information for each person's genotype. So it basically has all of the three-dimensional information, so columns, rows, and entries. So all of that information, you have to link in your analytic framework, and you have to deal with that information in a genomic analysis, right? And each columns has the dimension has their own information into their row and columns, right? So in a column table, you have the sample information across the rows and just the samples, and the information related to the samples across the columns, right? And if you look at the row table, then you have the variant information, variants across the rows, and information or annotation related to the variant across the columns, right? An entry table has the genotype information, and that is the same as the variant. Then this is the represent the genotype field, right? So all of that information you have to coming up with more efficient way. So if you only do sort of this analysis by the previous framework, like an Excel not gonna work in this Precept file. And if you want to work in a just native computational programming, like just Python or just R, and this thing's not gonna work. So that's why we are coming up with specific program called HAIR. Then knowing this program, I mean, of course we're not doing these HAIR things on hands in this semester, but I'll just explain you how the field use the latest technology, because in the near future you're probably in a position where you can handle the large amount of data set. If you become researcher, if you become clinician, if you become industry worker, in any situation there is no, I mean, it's inevitably you're gonna use the larger amount of genomic data set, and everyone probably taking advantage of high dimensional data set from clinics or public sectors, right? So that case, I would say almost the only solution to look up the genomic data set is HAIR, right? So so that's why I introduced the HAIR and their concept, and also want to emphasize that the VCF information is not just a single dimension. It actually come up with the multiple dimension, and you have to look up certain genomic information, look up certain the interpretation in the context of having this multiple dimension data set inherent in a VCF file, right? So I mean, if you have the burning desire in biological data science or genomics, I mean, you can go to the HAIR, then you can look up some documentation and see how, I mean, what we can do with these tools and in your experiments. Okay, so that is sort of the just a very brief update of how we analyze the genomic data set in the real world, and we learned about quite wide range of the terminology related to the genomics and bioinformatics. But knowing the terminology is the first step. If you understand the concept underlying the each process and analysis, then the terminology will be quite interpretable in the sense of the genotype identification and variant calling. But if you just try to remember or memorize the terminology bluntly, then I mean it's going to be very tough. So please keep in mind that please relate the concept from the Illumina sequencing and to the FASTA-Q and alignment and variant calling. Then all of that terminology will be, tightly intertwined and will be, quite digestible in your study. So, and again, if you have the questions or unclear things, then always welcome to leave yours on Slack channel. And then if you notice about the schedule, is we are finishing the session 3 by today. So we are continuing to have the review session on next Monday and also Q&A. I'm not sure how many things will be coming up over the weekend on the Slack. So it depends on your time. And anyhow, we'll have the review session on next Monday. And the exam for the session 4 will be next Wednesday, right? So, I mean, always the same format. And then we'll continue to have the session 5 on the following week. I don't know why this thing's not updated yet, but I thought from. Wow. Okay. So the schedule on the session 5 is actually changed, but yeah, I will check. Then will notify the update version. Okay. So I think all good and all things done, and hope you enjoy the rest of the week and also enjoy your exam preparation. And we'll continue to discuss on Monday.